you, dear viewers, dear students, dear old people, each and everyone. I welcome you to my YouTube channel. My name is still Osbaten Warinda, your biology teacher. In this episode, we are going to look at nucleic acids, and in these nucleic acids, we are going to look at very many other things which all fall under nucleic acids. So please make sure you follow me up seriously, because after nucleic acids, we are going to look at very many other components under nucleic acids. So under nucleic acid, in this episode, in this video, we are going to do much of the introduction. Then in other episodes which will keep on coming, still about nucleic acid, we are going to be looking at more details. So today we are going to deal with the nucleic acids. Some of you could be asking yourself that, teacher, what do you mean by nucleic acids? Nucleic acids, like they here, they are acids. And this nucleic acid also comes from the word, this word nucleic, is from the word nucleus. So these are acids which were first discovered within the nucleus. So that's why they are called nucleic acids. And these acids are very long molecules. I can call them polymers. In other words, the polymer is a very long molecule. So these are very long molecules, like a molecule which is too long. But this long molecule within a cell is very important as far as controlling all activities within a cell. So it means if I thought it's controlling all activities within the cell, it's another way of saying that it controls all activities that take place in our bodies. So these are very long molecules, or very big molecules, long molecules, which control whatever takes place in our bodies, which you can also take it to the whole body, that they control activities that take place in our bodies. Still, some of you could still be contemplating about these nucleic acids. I know most of you could have heard about DNA. Yeah, I think when I talk about DNA now, I can see you are smiling. At least you have heard about DNA. Whenever there's a wrangle between maybe a father and a mother about whether a given child is really a true member of that family, they usually go for a DNA test. So that DNA you hear is actually a nucleic acid. In fact, nucleic acids are of two categories. So we have got the categories, or you can call them types of nucleic acids. These categories is where we are going to find DNA, which I know most of you at least have heard. You may not be having a lot of knowledge about DNA, but at least I know you have heard about it. So that's the reason as to why I'm going to help you to expound more about the DNA in this new series of nucleic acids. Then another one is RNA. So these two are all nucleic acids that we are going to look at. So when I talk about nucleic acids, I'm talking about these two, DNA and RNA. I have said these were first discovered within the nucleus of a cell. However, some of them, though they are manufactured from a cell, they later they are manufactured within a nucleus, but they later cross the nucleus and come into the cytoplasm. But they were first discovered into the nucleus. That's why we are calling them nucleic acids. So after you have known that you have got these two, I know you might ask yourself right now, what is DNA in full, what is RNA in full, and so on. But before that, before I tell you, I will request that you hold a bit. We are going to discover, actually we are only going to discover it as we proceed. Now, nucleic acids are made up of, remember we have said this is a polymer. A nucleic acid is a polymer, is a long molecule. I mean is a long molecule. They are made up of long strands. The molecules are too long. So anything which is a polymer must be made up of smaller molecules, which we call monomers. So since we have got this one as a polymer, it means we have got monomers. So what are the monomers? What are the monomers of nucleic acids? For those ones who might not be much into the science, a monomer is a smaller molecule which adds up together to form a polymer. So since we have said that nucleic acids are polymers, it means they're made up of monomers. So those smaller molecules which add up together to form a polymer nucleic acid are called nucleotides. They are called nucleotides. So nucleotides are the small units that add up or that combine to form our polymer, which polymer we are calling nucleic acids. I hope we are together there. Nucleotides are simple molecules, are monomers, which add up together to form our polymer, which we are calling nucleic acid. So in this introduction, we are going to expound more on these nucleotides. What are these nucleotides? How are they? What are the components in the nucleotides? Then, as we go on, we are going to see how these nucleotides combine up to form our nucleic acids. So, our next subheading is going to be about nucleotides. So we are going to look at the nucleotides. So these monomers we are calling nucleotides. These small molecules which add up and form nucleic acids are also made up of some other components. So these components are the components of nucleic acid, of nucleotides are so we are saying this, though they are the small units, but these small units are also made up of some other substances. So there are three. One is a phosphoric acid, or you can also call it a phosphate group. That is one of the components of the nucleotide. Which nucleotide is a smaller molecule which will add up to form nucleic acid. Another component of nucleotides are we have what we call organic bases. But these bases contain nitrogen. So at times they say nitrogenous organic bases. So at times we can also call them like this, that they are nitrogenous organic bases. Then number three, another component of nucleotides, number three, and our last one, because I've told you there are three, the third one is what we call the pentose sugar. So nucleotides are made up of three things. One, we are saying they are made up of phosphoric acid. Number two, they are made up of organic bases which contain nitrogen. We can also call them nitrogenous organic bases. Then number three, we are saying pentose sugar. So we are going to look at one by one. Let me begin with the phosphoric acid. If I may begin with the phosphoric acid, let me come here. If I may begin with the phosphoric acid, Now for phosphoric acid, its structure is how the structure is. We have got the phosphoric acid. A bit. Phosphoric acid, its formula is like this. So when you are doing its structure, it means the structure are going to have phosphorus as the central atom. Then we can have structure which is like that. In case, because I've said it is a phosphoric acid or a phosphate, in case you want to write down for the case of the phosphate, because you have said phosphoric acid or phosphate group. So for the sake of the phosphate group, I hope you know very well the phosphate group is written in this way. Yeah. So in that particular case, it means we shall have our phosphorus as a central atom. Then we have then we have such kind of structure like that. So this is the phosphate group. So in terms of structure, for the case of the component which we have called the phosphoric acid, this is how it looks like. So the phosphoric acid is also one of the components of the nucleotides, which we are seeing, which is like this. Then another one we have seen is what we call the pentose sugar. So let me now take you through what we are calling the pentose sugar, because we have seen it's also one of the components. So the pentose sugar. Whenever you hear the word pentose, penta means something which has got 5 something. So this pentose sugar, it means that this sugar is made up of 5 carbon atoms. In fact, when you come and look at the structure, we have got these structures here. I have got a structure here, which is like this. I have got a structure which is like this. This is 1, 2, 3, 4, 5 carbon atoms. Then these 5 carbon atoms are arranged in this way. We have an OH there, H. We have OH here. We have H. We have OH here. We have H. Of course, here we shall have an H here. Then here we have got a molecule like that. So this is one of the pentose sugar. Now, one thing you need to know is that as far as nucleic acids are concerned, this sugar which has got this kind of molecule, this kind of structure, as far as sugars are concerned, a sugar which has got such kind of structure, we call it ribose. We call it what? We call it ribose. So a pentose sugar, a sugar which has got 5 carbon atoms, and it has got this structure, we call it ribose. Now, I told you to wait a bit because I was going to give you the something different which distinguishes DNA and RNA. So, this ribose, we are saying this ribose is our pentose sugar, which looks like this. But we have got two ribose sugars. How are they? Now, apart from me having this ribose which looks like this, I have got another ribose. Now, take note. This is carbon atom number 1. This is carbon atom number 2. If I take this one to be my center. This is carbon atom number 3, carbon atom number 4, and carbon atom number 5. So I have got another ribose sugar. I have got another sugar which is almost the same structure. But for it, it is lacking an oxygen. It is lacking an oxygen on carbon atom number 2. And this is the one which will look like this. So for it, it looks like this. You have got a sugar which is O, like this, also made up of 5 carbons. But now, in this case, you have the OH there, you have the H. Why I'm doing like this? Because this carbon is supposed to be surrounded by 4 bonds. Likewise, but now when we come to this carbon atom number 2, carbon atom number 2, instead of having an OH, I have just an H and an H. Then the other ones are the same. Or H here, I have an H there, there's an H here, CH2, or H. So the only difference between, now still this is my first carbon atom, number 1, number 2, number 3, number 4, and number 5. All these ones are ribose sugars. But I'm saying, when you look at these two structures, there's only one difference. The only one difference is that we have seen, apart from this, the only difference between this one and this one is that for the case of this, instead of having an oxygen here, which is here, this one lacks the oxygen. And because it is lacking the oxygen, then this ribose, we are going to give it another name, and we are going to say it is deoxyribose. Deoxyribose. Why deoxy? We are saying deoxy because on carbon atom number 2, it is lacking an oxygen, which the ribose is having. So these are the two pentose sugars we have. And it is from these two pentose sugars where we get the two types of nucleic acids. One being the DNA, another one being the RNA. So the major difference here in terms of writing this in full is that this DNA in full is deoxyribonucleic acid. Deoxyribonucleic acid. Then this one here, now we are giving it deoxyribo. This is the DNA. Why deoxyribo? It is called deoxyribo because when you look at its nucleotides, when you look at the nucleotides which make up this DNA, those nucleotides that make up DNA, their pentose sugar is deoxyribose. I hope you are following me very clearly. This is deoxyribose. Deoxyribonucleic acid because when you look at the nucleotides which make up this DNA, their pentose sugar is made up of, their pentose sugar is called deoxyribose. Why? Because it is lacking an oxygen on carbon atom number 2. So it means you can actually easily tell me what RNA is in full. So it means RNA in full is ribonucleic acid. Why ribonucleic acid? So it means you can also now tell me. So it means the ribonucleic acid is because when you check the nucleotides which make up RNA, the pentose sugar which is found in the nucleotides that make up RNA, that pentose sugar is none other than ribose. So it means we have got, in terms of naming, this is called DNA or deoxyribonucleic acid because it has the deoxyribose pentose sugar. Then this is the RNA ribonucleic acid because the pentose sugar is none other than ribose. So this word ribo is coming from the short form of ribose. Likewise, even this is supposed to be deoxyribose. So we just make it deoxyribonucleic acid. I hope we are now together there. So, after you have understood that, remember we said we have 3 components. So we have finished looking at the structure of the phosphoric acid. We have now finished the pentose sugar. So it means the next thing we are remaining with is the nitrogenous bases. So my next part here, we are going to look at another component of nucleotides which we are calling nitrogenous organic bases. So how are these ones? Now, as far as these bases are concerned, we have got in each nucleic acid, let it be DNA or RNA, we have got 4 organic bases. And these 4 organic bases are, so I can say these include, one, we have got we call adenine, which we usually we also symbolize using A. Another one is guanine, which usually use G to symbolize it. Then another one is cytosine, which we usually indicate using capital C. And then another one is thymine, thymine, which we signify, which we symbolize using capital T. So, but we have another one. I've told you there, in each and every nucleic acid, nucleic acid we have got 4. But surprisingly, I'm going to put here 5. I know you're going to ask yourself why. So we have another one. Another one is none other than uracil, which uracil we also indicate using U. So I have said we have in each and every nucleic acid we have got 4, but I am writing 5. I know you are saying why. The reason is very simple, that thymine and uracil, these two interchange. How? Thymine is found in DNA. But when it comes to RNA, this other nucleic acid, instead of having thymine, the thymine is replaced by uracil. So this one is found in RNA. So that's why I'm saying that in each and every nucleic acid, we have adenine, guanine, cytosine, and if it is DNA, we have got thymine, but if it is RNA, the fourth one is uracil instead of thymine. I hope now that one is clear. Okay, these bases are of 2 categories. They are categorized into 2 forms. So we have got the different categories. We have got the different categories of the nitrogenous bases. These bases are listed up there. They are categorized into 2, and those categories are: 1, we have got a category which we call the purines, and then we have also got a category we call pyrimidines. So these 5 bases are categorized into 2. We have got what they call purines and we have got what we call pyrimidines. What's the difference between these 2? The difference between these 2 is in their structure. Now, with the purines, these are organic bases whose structures are made up of. So these ones, the structure consists of 2 rings, and these 2 rings, one of the ring is a hexose ring, and then the other is a pentose ring. You find that their structure is in such a way that they have a 6-sided ring, but at the same time you have a 5-sided ring next to there. 1, 2, 3, 4, 5. So the structures are in that form. So those are the ones you call the purines. So the purines, their structures are in such a way that they are made up of 2 rings, and these 2 rings, one is a hexose ring, which is made up of 6 sides. Like you see, the way you see this, so these are 6 sides, and then in touch with another ring which is made up of 5 sides. We are calling the pentose ring. Like the way you see this other one, which is now 1, 2, 3, 4, 5 sides also. Then for the case of let me give now. What are the examples which fall under, out of these 5? What are those examples which fall under purines? The examples which fall under purines are these 2, and these 2 are adenine and guanine. Those are the 2 examples. In other words, when I come here, those two, the adenine and guanine, are all what you call purines. If you want to remember, I can give you a small technique. If you want to remember that it is adenine and guanine which fall under purines, I can give you a small technique. A small technique is by you saying purine saga. Purine saga. The AG you are adding here, the AG you are adding there is signifying adenine and guanine. Yeah, That way you can be able to remember. So the adenine and guanine fall under the category we are calling the purines. The category of bases whose structure are made up of two rings, one being a hexose ring, a six-sided ring, and then another one being a five-sided ring, which we are calling the pentose ring. And that's what we have. When you check here, if you check here, this is how the adenine structure looks like. Are you seeing? So you see this is a six-sided ring attached to the five-sided ring. So that is how the structure of adenine looks like. Then guanine, you also see it is six-sided and then five-sided. And then that's how it is looking like. So that's what we mean by saying purine. So it means for the case of the pyrimidines, for these ones, they are made up of one ring. Their structures have one ring, and this ring, the ring is a hexose one. Hexagon usually means something which has six sides. So the ring is hexose, meaning that the ring is six-sided. The ring is six-sided. So when you check, when you come and check, so it means if we have said that adenine and guanine are the only ones which are falling under purines, and we have 5 of them, so it means the other remaining ones, the other remaining bases, all fall under the pyrimidine. So you can give me those bases. So those bases are, so it means here the bases we have here are, so tell me, so we have C, that is cytosine, then we have got thymine, T, and then we have also got uracil. So it means these other remaining 3 are all pyrimidines, and I've told you that their shape is a six-sided shape. As the beta shapes can be seen here. When you check with me here on this part here, so here I'm giving you the shapes of the different pyrimidines. So you see this is cytosine, that's how its shape looks like. Then this is the shape of thymine, that's how it looks like. And then this is the shape of uracil. So that's how it looks like. So all these ones are all pyrimidines. So those are the different components that make up the nucleotides. So before I can end my video, I cannot fail to tell you, to show how these components of nucleotides combine together to form, how these 3 combine together to form a nucleotide. So that one I cannot fail to show you. So allow me rub this end, and now I show you how these 3 combine to form a nucleotide. Okay? So our next question we are answering is that how do the components we have already seen, the components add up to form a nucleotide. Add up to form a nucleotide. So I will pick up. So what I'm going to do, I'm going to say that let the phosphoric acid, a phosphoric acid be, let me symbolize using that. Then I also let the pentose sugar be symbolized by something like that. And then the organic base or the nitrogen base be signified by that. So what is going to happen is that these 2 are going to combine in such a way that we shall have our pentose sugar there. Then here it will combine with a phosphoric acid. Then at this end it is going to combine with a base. So this will be our base, nitrogen base. This will be our pentose sugar, and then this will be our phosphoric acid. Now, can we put real things and we see how this one is formed? Now, all these combine. When they combine, there is a loss of water. So these three combine. So the three components, I can put it that the three components combine with loss of 2 water molecules. With loss of 2 water molecules in a reaction called this reaction we call it a condensation reaction. A reaction where we lose water molecules, we call it a condensation reaction. Now let us put real substances. Instead of putting this symbol, let me put the real things, and then we see how this reaction happens. How do these three combine to form a nucleotide? Now, if I pick my phosphoric acid there, so I will have something like this. I will have something like this. P. I have something like that. I have it there. I have this one. I have that one there. Please follow me very well. I have something like that. Then after I bring my base, I bring my pentose sugar. Let me pick a pentose sugar maybe for ribose. So it means now I'm going to form a nucleotide of RNA since I have picked a ribose sugar. So I picked a ribose sugar. So the ribose sugar is here. I have that like that. I have my H there, my OH there, OH there, H, my H. Then I have CH2 there, and I have OH there. Yeah. I hope this is correct. I think I'm missing the hydrogen there. Are you seeing that? Then after that, I bring a purine, or I bring my base. So I'm going to put a base here, but I want you to come with me here. When you check on all these bases, at least for each and every base, I am sure that you can see there. I hope you can see there a part which has got something like this. I hope you can see a hydrogen which is in this way. For example, if I come here on this, you see I've got this bond here, where I have a nitrogen and I have an H, and then I have these two bonds. If I come here, you see I have got this A, I have got this H, I have got a nitrogen here and the two bonds there. If I come here, you see I have this, I have got my N and I have two bonds here. Are you seeing? If I come here, I have got my H, I have got my N, nitrogen, and then I have got those two bonds, which is the same as this. So likewise, even here, you see I have got this area here where I've got an H, a nitrogen, and then the two bonds. So I will request that you allow me, instead of using any of this, I just pick out this point here, that at least for each and every base I am seeing such kind of, where I have a hydrogen, a nitrogen, and this nitrogen is combined to two other bonds to the ring. So instead of putting all the other details, since I don't have enough space here, allow me put just that. So if I'm having my, I think I have H, I'm having H, then N, then this N is connected to the ring. That is now the bonds to of the ring. Let me do like this. So when we are going to form a nucleotide, this is what happens. What happens is that this OH here and this H of the phosphoric acid combine. So we shall have these two combine. Take note, the oxygen which is remaining outside is the oxygen of the phosphoric acid, not of the pentose sugar. So it is the oxygen of this pentose sugar and the OH with oxygen of the pentose sugar and then the H from the phosphoric acid which are combining. So when these two combine, they will form the ions which are going to form our water molecule. So that is one of the two hydrogens and then the oxygen. That's our first molecule of water. Remember here I've told you that we lose two water molecules. So our first molecule is that one. Then another molecule is here. The OH. Remember this is my carbon atom 1, carbon atom number 2, number 3, number 4, and then number 5. So on this carbon atom number 1, the OH here, this OH will come and will combine with this H from the base, and this is going to form another second water molecule. So it means once these two combine, it's where now you will end up having something like this. I hope you are following me very well. So that's why you end up having something like this. So you have got a phosphoric acid here, like that, like that. But now down here you have got an oxygen. You see this bond here, oxygen. Now this oxygen is combining with a carbon atom here, which is carbon atom number 5, which has got its two hydrogens there. But this carbon atom is combined on carbon atom number 4, which is part of the ring. So you have got that ring like that, like that. You have an H here. This is OH. This is my H here. Then this is OH. Hope you are following me very well. Then this is an H. Then I have got an H here. But in addition to that, I have got this. Remember, this OH was attached to this carbon atom. So I have got another bond. This bond is connecting to a nitrogen, which nitrogen is joining up with the ring, the other side, with the base, the other side. So you see, I have got a bond formed here. I have got another bond which is formed here to form. So this one I have formed here. Since this one is a phosphoric acid, this was a pentose sugar. This pentose sugar is now ribose, and then this was a base. So this one I have formed here is what we are calling, I'm calling a new. So this one I have formed here is a nucleotide. That's what you call it, a nucleotide. So this bond which has been formed here, we call it a phosphor. We call it a phosphodiester bond. Called a phosphodiester bond. So in that way, the phosphoric acid, the pentose sugar, and the base combine through a condensation reaction which involves loss of water molecules, and they form the nucleotide. So it is this nucleotide which will combine one by one, and they form what you call a nucleic acid. So after we have understood how these three have combined to form a nucleic acid, allow me a few minutes to conclude in this way. This is like this. I also request you to allow me to grab this area here. So what will happen is that if I have a nucleotide like that, and then I have another nucleotide which is here, and then I have another one down here, like that. So these are all nucleotides. Remember, I used this example here at the beginning, which we have seen it is like this in reality. So if I have very many nucleotides, So these dinucleotides will combine to form a very long chain. A very long chain of what? A very long chain of nucleic acid. So it means that if I have this nucleotide, I have another one down here. Let me add another one also. Have another one here. So all these will combine to form a nucleic acid. How? Let me extract one. Let me extract one thing here. So it means this carbon atom, remember this was a carbon atom number 1, number 2, number 3, number 4, number 5, number 5 somewhere in the middle there. So we shall have a bond between, we shall have a bond which will be, we shall have a bond in between here. We are going to have a bond in between here. Then we are also going to have another bond which is formed in between here, and then we shall have another one in between here. And then we are going to be forming a very long chain. How? For example, I will come and I put that bond. Let me indicate it there. So remember, this is a phosphoric acid. So we shall have a bond there. We shall have another bond here. We shall have another bond here. So all these bonds still are, these bonds which are going to be formed here are all going to be phosphodiester bonds. Are going to be phosphodiester bonds. Let me also wrap something small here. And I put a real molecule. For example, if I have something like this, if I have something like this, like that, then like that, we have seen this one is going to combine with the, I'm copying out this one here. Allow me just take it like that. Then I have got a base here, and then I have, remember here I have a carbon atom which is the OH, which has an OH here and an H there. Yeah, allow me just simplify like that. And then I have another one here. Which is the same. So this is one nucleotide. The whole of this one is one nucleotide. So I have another one which is still the same, still the same. Allow me like this. Allow me like this. So we have something like that. Then I have CH2 there. This one is combined to another carbon atom here. It's combined there, like that. And then I have a base here. Allow me just simplify in this way. Now, what happens is that now you have one nucleotide. This is one nucleotide, and this is another nucleotide. So these nucleotides should combine to form a very long chain of a nucleic acid. So what will happen is that likewise, if you have a nucleotide, another nucleotide here, the same thing down here, what is going to happen is that this oxygen, this OH on carbon atom number 3 will come and combine with this, will come and combine with this H here, with that. So it means here we shall have a loss of water molecule, because these two are going to, so when this one, when this OH combine with this H from the phosphoric acid of another nucleotide, you will form a bond, which still is going to be a phosphodiester bond. So that is a bond I am showing you here. So this bond is that one. This is a phosphoric acid, but which is attached to this pentose sugar and that base. And what has happened here can happen down here, and at the same time can happen up there, leading to the formation of something which is as long as this. So if you have this one as a nucleotide, you have also this one as a nucleotide, you have this one as a nucleotide, so you see that these nucleotides are combining. This one combines with this one. And how do they do that? I have shown you. If this is your pentose sugar with an OH here, this OH combined with the H from the phosphoric acid of another nucleotide to form a bond. So that bond is this one you are seeing here. So what is happening here is going to happen here, and it's also going to happen up there, and in that way you form a very long chain of nucleic acid. I cannot exceed here. So in the next series we are going to now become specific, and we are going to be looking at the specific nucleic acid, that is RNA and DNA. You don't need to miss that. So I am very sure that you have learned a lot. So please make sure, in case you're watching for the very first time, make sure you subscribe, and please wait, be anxious for the next video where we are going to expound more on the real nucleic acids. I thank you very much.